As I mentioned, today's speaker is Mike Lin. I actually know Mike because we use the excellent but somewhat obscure programming language OCaml. So I posted a question on the OCaml list that made it clear that I was using OCaml for phylogenetics and got this response back that was something like, really? Because he was doing the same, and he probably didn't expect anyone else to be doing that. Anyway, looking into his work, I found that he was doing really awesome work doing using codon models to find genes and other interesting genomic features. So I thought I'd invite him to speak in the seminar. And Mike is a graduate student at MIT in Manolis Kallis' lab. So what year are you, Mike?
It's a little complicated. I'm hopefully graduating soon and prefer to count down.
Right on. We'll take it away.
All right. Thank you, Eric. I'm Mike, and I'm presenting today some work I've done over the last few years on using phylogenetic codon models in genome alignments of many species, particularly mammalian species. And the goal of this work is to locate protein coding regions that are actually, in addition to coding for an amino acid sequence, also performing some other function, and as a result is under sort of dual selective constraints to preserve the amino acid sequence throughout evolutionary time on the one hand, but also to perform some other function, and we'll explore what some of those are. So to introduce just the context and how we look at the alignments here, what I'm showing is a schematic of a genome sequence alignment. So we have the fully sequenced genomes of 29 different mammalian species here, human near the top, and all the way including dolphins, mice, rat, cow, bats, shrews, elephants, and so forth. And these genomes were sequenced in the 2005 to 2008 timeframe. And I'm showing a particular region of the genome, which is the 3' end of the open reading frame of the CFTR gene, a very famous human gene linked to Mendelian disease. In particular, what I'm showing, so it's the very 3' end of the open reading frame. So this purple column in the middle is actually the stop codon at the end of the open reading frame of that gene. And, oh, okay, I was a little zoomed out here, actually. Much better. Okay, so the middle column here, the purple column, is the stop codon of that gene. And what we're showing in this schematic, you can see it's very colorful, is at the very top row is an inferred ancestral sequence. So that's sort of what we infer based on the extant sequences the mammalian ancestor held. And the colors are indicating substitutions of codons with respect to that inferred ancestral sequence. So the white codons are identical to the inferred ancestral sequence. The bright green codons are, the bright green, excuse me, codons are synonymous to the inferred ancestral. The dark green are non-synonymous but conservative of amino acid properties. Other non-conservative codon substitutions are red. And then we're also showing in orange frame-shifted regions, and in the 3 other colors are 3 stop codons. So what we can see is that upstream of the stop codon of this very well-known and well-conserved protein coding gene is upstream, we see this sort of tremendous bias towards synonymous and conservative substitutions with respect to that inferred ancestral, exactly as you would expect, given that this sequence encodes a conserved protein. Okay, so over evolutionary time, since mammalian divergence, non-conservative and other sort of likely deleterious substitutions have likely arisen, but were selected out. Now, downstream of that stop codon, so over to the right-hand side here, you can see the sort of opposite pattern. Okay, instead of this tremendous bias towards synonymous and conservative substitutions, we have frame-shifted regions, as well as a lot of non-conservative substitutions and nonsense mutations, substitutions to stop codons, and so forth. So if you flip through a lot of pictures like this of protein coding genes throughout the genome, this is pretty much what you would expect to see, right, given that most protein coding genes are very well conserved across mammals. And indeed, it's exactly what you do see. In particular, their synonymous substitutions tend to be pretty well tolerated over evolutionary time. So if you've done, so everyone in phylogenetics, I'm sure at some point has made the assumption that synonymous sites are neutral in evolution, and to a first order approximation, that's true, and we see that reflected in these alignments where we see synonymous substitutions largely tolerated. In this work, we're interested in looking in situations which that's actually not the case. So as a motivating example, I want to show just this a fascinating result from a biology lab a few years ago. So this takes place within the mouse HOXA2 gene. And the finding in this paper was that there's a portion of the coding sequence of HOXA2. Okay, so here is showing the second coding exon of HOXA2, and then zooming in and showing different regions within that open reading frame broken down. And the finding of this work is that if you take part of that coding sequence and put it upstream and recombinate a mouse and put it that portion of the coding sequence upstream of a reporter gene, that coding sequence actually acts as an expression enhancer and drives tissue-specific expression of the reporter gene in the developing mouse hindbrain. Okay, so this is a sequence which, on the one hand, as part of HOXA2, is encoding the very important, presumably HOXA2 protein sequence, but additionally seems to perform some capacity as a tissue-specific expression enhancer. So it's a very interesting situation where, again, you have this sort of dual information encoded of encoding a protein sequence and also, in this case, acting as an expression enhancer. And zooming in on the sequence, what we actually see is that there are transcription factor binding sites embedded within that coding sequence, so RTE and SOX2 binding sites in that region. So we were very fascinated by this, and we wanted to study sort of the evolution of this this sort of phenomenon for this example certainly, but also throughout the genome as well, and find out, well, what other sorts of things are going on embedded within protein coding regions. So I'm going to now show sort of just a teaser of where we're going with this. So this is a genome browser diagram of this gene HOXA2, so the upstream exon and this downstream exon. And well, I'm showing something, a track we generated, which is the local synonymous rate estimate. And I'm not currently saying anything about how we're computing that. I'll get to that. But what it's showing is in a pretty short window of about 9 codons, it gives us an estimate of the synonymous substitution rate in that short window relative to the average for the entire open reading frame. So there's some average synonymous substitution rate across the length of that open reading frame. And this is showing now within a short window what is the substitution rate in that window relative to that average. So a measurement of 1 says it's about the average, and a measurement close to 0 says that there seems to be a greatly reduced synonymous substitution rate in that window. So in the second exon here we can see the dip in the synonymous substitution rate is the region corresponding to the tissue-specific enhancer, which I had shown on the previous slide. So again, if you totally cut that region of DNA out of the protein coding gene and put it upstream of a reporter gene, that is adequate to drive expression of the reporter gene in a specific segment of the developing mouse hindbrain. You can see also, I'm sure you notice there's a second dip in the synonymous substitution rate at the very 5' end of the open reading frame for this gene. And it turns out that in a sort of separate independent study, that region was also shown to drive tissue-specific expression in a different segment of the developing mouse hindbrain. Okay, so a very fascinating situation. We have a HOX gene here, so obviously gene these Hox genes are key in the specifying the animal body plan, including the developing nervous system. And it seems that not once but twice, in two different places, embedded within the open reading frame, there is a tissue-specific expression enhancer, potentially regulating the expression of this gene or perhaps other nearby genes. That's still under investigation, I think. I'll now sort of dive into getting a handle on how it is that we are measuring this synonymous substitution rate across mammals in pretty short windows. So just to give you a feel, to go back to the genome alignments, I'm now showing you snippets of the genome alignments. I'm showing you little 9 codon windows again, and 5 of them, 1, 2, 3, 4, and 5. They're snippets out of the open reading frames of 5 different pretty famous genes. Aldehyde dehydrogenase, CFTR, which I showed before, BMP4, another very important transcription factor, GRIA2, and HOXA2. So obviously I'm not showing you the whole open reading frames, but just little 9 codon windows snipped out of those open reading frames. Now, you can sort of measure across the genome, across all genes, and find that on average, a typical codon site across these 29 mammalian species shows about 4 synonymous substitutions. So that's what's being illustrated in the 2 windows on the left from aldehyde dehydrogenase and CFTR, where again, the top row is an inferred ancestral sequence, and the bright green boxes are synonymous substitutions with respect to those that inferred ancestral sequence. So again, typically on average, given a given column, codon column of this alignment, we see about 4 synonymous substitutions. Now in the 3 windows on the right, okay, where the rightmost is that downstream enhancer in the HOXA2 that I've been talking about, it seems like we're seeing considerably fewer synonymous substitutions per column than that average of 4, right? So somewhat reduced in this 3rd one, reduced more in GRIA2, and in that HOXA2, it seems there's essentially no synonymous substitutions exhibited here. So it seems like that's potentially interesting, right? And potentially this provides us a strategy to go out and look for these overlapping functional elements. But if you think a little bit about now sort of developing a quantitative method to look for these, it gets a little complicated, right? So how much is it reduced, right? It seems we have 3 different degrees of reduction here. How do you put a number on that? Okay, now, and then suppose you look at a window and you find you've developed some way to measure synonymous rate, and you find that in fact it's reduced. How would you say that reduction is in some way statistically significant or not a result of sort of random variation in synonymous substitutions that you could explain by some uninteresting random process? And then if you think about it even harder, you can see that actually the problem is quite intricate, right? So in particular, suppose I'm not sure there are any in these particular alignments, right? But suppose we have a codon site that encodes a methionine or tryptophan, ATG and TGG, and so those are non-degenerate codons. Namely, there's no other codons that are synonymous for either of those. So if you have a conserved methionine or tryptophan site, right, then of course you're not going to see any synonymous substitutions in that site, and it's not surprising that you don't see any synonymous substitutions because no such substitutions are even possible. And so that's an extreme case, right? But in general, the different amino acids have different degeneracies with the codon. Some are 4-fold degenerate, some are 6-fold degenerate, some are 2-fold degenerate, and so forth. And you can imagine, right, you would want to really consider that when you're developing a way to measure how much is the synonymous rate reduced, and is that synonymous reduction significant. So hypothetically, right, imagine you had a window that was only methionine and tryptophan residues. I don't know if there's such a window, but just hypothetically imagine there was. Then it would be totally unsurprising to see complete conservation of that window with respect to the synonymous rate because there are no synonymous substitutions possible. So we'd like to develop a method to sort of assess this in a way that takes that all into account in a hopefully elegant way. So to introduce that method, I will sort of appeal by analogy to a sort of a very similar but simpler problem, namely the use of phylogenetic models to measure evolutionary constraint at the nucleotide level in genome alignment. So if you use the UCSC genome browser and you've noticed that they have this track that's showing conservation by phylop. So what I'm going to describe now is sort of how that method works. And there are a few other similar methods like SCOAN or GERP that are based on the same principle. But the basic idea is, again, these are operating at the level of nucleotides, so not considering anything about the genetic code or codon structure. But they have a phylogenetic model, okay, which uses the sort of typical approach in these likelihood-based phylogenetic rate models where there's a presumption that evolution is a continuous time Markov process happening on the phylogeny. So we have a tree with the topology and branch lengths, and we have a 4x4 nucleotide rate matrix that specifies the instantaneous rates at which each nucleotide changes to any other. Okay, and then together these things, this tree and this rate matrix, this phylogenetic model determines a probability distribution over alignments. So if you have any given alignment or even a single nucleotide column of the alignment, this model will tell you, well, that has a certain that alignment has a certain probability relative to all other possible alignments or columns. So in these nucleotide level constraint detection methods like phylop and GERP and SCOAN and a few others, we have this model which is meant to represent the evolution of neutral sites. So they're often estimated by maximum likelihood from ancestral repeats or fourfold degenerate sites, but that will get a little uncomfortable in this talk. But in any case, there's this nucleotide level phylogenetic model, which is estimated from presumptively neutral sites and is meant to sort of represent the evolution of those sites. And then there is then an alternative model. So when we now want to study an individual column of the genome alignment or short window in the genome alignment, we take that null model and we have a free parameter rho, okay, which acts as a scale factor on the branch lengths in the phylogenetic tree. Okay, so we have our null model, and we now study an individual column of the alignment or a little window of sites, and we estimate this parameter rho by maximum likelihood. So again, there's the phylogenetic model determines a probability of the alignment, and we're now going to adjust the assumed substitution rate throughout the phylogenetic tree in such a way as to maximize that probability of a model, the probability of the column or the alignment that we are studying. Okay, so suppose we do that and we find a very reduced rho. Then that would sort of suggest that there's been a very low rate of substitution in that alignment, and in turn then suggests that there has been some purifying selection on that region. Okay, so that again, these are very widely used methods. And if you use the UCSC genome browser and look at the conservation track, this is exactly the type of method that is used to generate that. So coming back now to our problem of looking for reduced synonymous rates, we're now going to extend this general approach, but instead of using a sort of 4x4 nucleotide level model, we're going to use a 61x61 codon level model. Okay, so we start off with sort of the same idea for a null model, namely we have a phylogenetic model, but which now models substitutions of codons rather than individual nucleotides. And we estimate it from some data in such a way that we believe it represents the evolution of typical codon sites, that is, sites without any unusual evolutionary constraints on their synonymous sites. Okay, so very similar idea. The same algorithms and everything are used to estimate the model. It's just that it operates on codons instead of individual nucleotides. It has very many more parameters and so forth. But with 29 fully sequenced genomes, we have plenty of data still to estimate all those parameters. Okay, and now, as our alternative model, instead of having sort of one free parameter that scales all of the evolutionary rates, we're going to have two free parameters. One which will scale the synonymous rates, and one which will scale the non-synonymous rates. Okay, so what that's going to do now is we're now going to study short windows of codons and estimate these two parameters by maximum likelihood. And that will actually allow us to distinguish reductions or changes rather in synonymous substitution rates from non-synonymous substitution rates. So the structure of this rate matrix and these parameters is such that the parameter lambda s will scale only the synonymous rates in the model, and the parameter lambda n will scale only the non-synonymous rates in the model. So if we take a small patch of the alignment and estimate these two parameters, and lambda n is kind of a nuisance parameter on amino acid level evolution, which we're less concerned with in this setting. But if we find a very small lambda s, then that's potentially interesting, right? It suggests that there's a small, there's a very low synonymous substitution rate. And the structure and the model is structured so that it will, in doing so, take into account what I mentioned on the previous slide about accounting for different degeneracy levels of the different codons and so forth. So the purposes of moving to this much more complicated codon level model is so that we can actually disentangle the effects of disentangle synonymous and non-synonymous substitutions when we study the alignment, and take into account the structure of the genetic code in terms of how surprising is it to see a synonymous substitution or not, given what's been encoded. And then I'll also just mention in closing for this slide that if you're familiar with the use of these phylogenetic models to study protein coding genes, you're probably familiar with the DNDS ratio or omega in PAML or any of these other packages. So we're obviously using related ideas here, but where that's sort of a ratio of non-synonymous and synonymous rates, here we are separately estimating synonymous and non-synonymous rates in absolute terms rather than relative to each other. Okay, so let me now come back to these 5 windows. Okay, so I'm sort of repeating a slide that I showed before, but I've added now the maximum likelihood estimate of that synonymous substitution rate relative to sort of the average of all protein coding sites. So again, the null model is supposed to represent typical codon sites without much constraint, much unusual constraint on their synonymous sites. We are presuming here that these effects of overlapping selection are relatively rare. So just by averaging over all sites, we'll get a pretty good handle on that desired null model. And to the extent that we're off, it will tend to be off in a conservative direction. But so as I said, the 2 windows on the left are more or less typical throughout the genome. The CFTR window is the maximum likelihood estimate is exactly 1. So that seems to be about average. And then we now have a precise sort of measurement of the synonymous rate in the 3 other windows where we had said it seems to be reduced. So in the BMP4 window, we seem to see about a 50% reduction in the synonymous rate. In the GRIA2 window, about a 90% reduction. And in the HOXA2 window, apparently 100% reduction. Okay, so we've made a lot of progress here. This is pretty cool. We have this pretty nice way now of measuring this synonymous rate. So let's go out and do this sort of throughout the genome. So we'll measure these 9 codon windows sort of across all open reading frames and take a look just at first at general genome-wide trends of those rates. So What I'm showing here is sort of an idealized gene model where I'm going to show averages for first exons, middle exons, alternatively spliced cassette exons, and last exons. And then I'm showing with these box and whisker plots sort of the genome-wide distribution of non-codon of the synonymous rate estimates in non-codon windows within each type of exon. So on the left of each group of three, I'm showing it sort of the distribution of that relative synonymous rate in the first 9 codons of each exon. Over on the right of these in the last 9 codons of each exon, and then in the middle, somewhere from the middle of those exons. So there are a few interesting things that sort of immediately stand out from looking at these distributions. We can see in all types of exons, there seems to be a somewhere between slight and substantial reduction in this synonymous rate at the 5' and 3' ends of the exon. So that's it seems, when you're averaging over all exons, you can see a significant reduction averaged over all exons, and that sort of makes sense if there's some sort of embedded motifs that are sort of regulating splicing activity. And in fact, there are this has been very well studied. They're called exonic splicing enhancers, and it's very well established that the coding sequence context near splice sites affects the efficiency of the splicing machinery. In the middle of middle exons, as you would expect, you have about the average rate. And then we see a much more dramatic effect in these alternatively spliced cassette exons, where very dramatically at the 5' and 3' ends of those exons, and also to a pretty large extent, even in the middle of those exons, we have these reduced synonymous rates. And so this is also consistent with there being additional regulatory elements embedded within the coding sequences of these exons, which regulate the alternative splicing of these cassette exons. Okay, so again, we're just looking at genome-wide averages here, but we see some very striking results, which is nice. To come back to the previous slide, remember there were 2 parts of what we wanted to develop a method to do. The first was to make this measurement, and we've now sort of done that and looked at the genome-wide trends, and they seem very striking. There was a second aspect of it, which was, well, when we see a reduction, is that reduction statistically significant? In other words, suppose we looked at some window and found lambda s of 0.92 or something. Is that interesting, or is that in fact something that's can be explained as part of the variance that arises from this underlying Markov chain that is the basis of these phylogenetic models, or just in any sense, is it explained just as uninteresting random variation? So to address this, we can and sort of build upon methods that are very widely used with these phylogenetic models. We will formulate a likelihood ratio test, which will basically map a sort of the probability distributions that we're seeing for the window and the estimated rates onto a p-value for the hypothesis that the synonymous substitution rate is reduced. So again, this, if you're familiar with how phylogenetic models are used with codons and DNDS and stuff, this should be pretty familiar. But basically, just to briefly go through it, as I mentioned, these phylogenetic models for any given window determine a probability of that window versus all other possible alignments that you could have seen. And we estimate our parameters lambda s and lambda n to maximize the probability reported by that model. Okay, so if we in the numerator here take the sort of optimum probability of that, where we allow the synonymous substitution rate to be reduced to 1. Okay, and we're sort of using the lambda n, the non-synonymous rate as a nuisance parameter. We sort of maximize it. We set it to its optimal value, but we're not so interested in it. Okay, so we take the ratio of that probability with the corresponding probability computed under the assumption of the genome-wide average synonymous rate, lambda s equals 1. Okay, so this is a statistics, or the ratio of these two probabilities. And there's some very useful but kind of obscure theory about this statistic, this likelihood ratio statistic, as to, at least in large samples, how it will distribute under the null hypothesis, namely sort of twice the log of this likelihood ratio can be shown if the null hypothesis is true, to distribute as a chi-square distribution with 1 degree of freedom. And there are various other slightly fancier assumptions you can make, but this one is simple and widely used. So having that null distribution allows us then to, well, when we have a window and we find, say, a very large likelihood ratio, meaning that adjusting the assumed synonymous rate allowed us to explain that window much, much better. We can now map that using this null distribution assumption onto a p-value. Okay, so what I'm showing here is a scatter plot of where each point represents one 9-codon window from somewhere in the genome, or the protein coding genome, that is. On the x-axis, we have this lambda s statistic. Okay, and it's a log base 2, scale goes 1, 1/2, 1/4, 1/8. So over to the left, we have very reduced synonymous rates. And then on the y-axis, we have the p-value for the reduced synonymous rate that we compute based on these assumptions. So as you would expect, as the measured synonymous rate gets lower and lower, you tend to see more and more significant p-values for reduction. Just to illustrate that there's a point doing all this, we can also see that there are a number of windows that get very low estimated synonymous rates, but in fact our model is telling us that those reductions are not significant. So a possible explanation for that again is, suppose there are some windows that are very heavy on methionines and tryptophans, or twofold degenerate codons, or regions where just the model would tell you not to expect very many synonymous substitutions. And then so we compute this measurement and we see, yeah, there are a few synonymous substitutions, but by doing this significance estimation, we can see that actually it's not very surprising that we didn't see very many synonymous substitutions. So to bring this together, and previously we had the ability to sort of measure genome-wide averages of this. What this likelihood ratio test allows us to do is now to give a yes or no answer for each individual window. Is there interesting evidence for reduced synonymous rate in that individual window, rather than looking at genome-wide averages? Okay, so that allows us to actually define, locate. So we can generate a GFF or bed track of, well, here are a set of synonymous constraint elements, as we call them, throughout the genome. So again, we have resolution for 9 codon windows in the initial work. We had also looked at 15 and 30 codon windows. Much later in the talk, I'll get to how we're doing more recent work to drill down even further. But what we find, again, by applying that statistical test throughout the genome, is where we locate more than 10,000 of these synonymous constraint elements throughout known human protein coding genes. You find at least one within more than a quarter of all known human protein coding genes. And together they cover about 2% of the synonymous sites in the human genome. And on average, they'll show 70 to 90% reduced synonymous rates. That corresponds to the lambda s of like 0.1 to 0.3. So just looking at some of these plots, in the upper left here, we have like the, just the genome-wide distribution of this lambda s statistic. So we can see it has this heavy left tail, which we're interested in on this. Well, it's on the log plot, but there's a heavy left tail. On then on the lower left here, again, we have this mapping of the measured lambda s to a p-value, and we apply the appropriate sort of multiple testing corrections to these p-values when we define these elements. And then an important point, again, how we define these was we assessed each window with respect to two different null models. One was the sort of genome-wide average null model, which I've been discussing up to this point. And we also assessed each window with respect to a second null model, which was actually tailored to a smaller genomic region surrounding the window of interest. So the idea here is there's it's well known that there's considerable variation in composition and neutral rates in different parts of the genome, arising from different recombination rates and other evolutionary effects. So it's important when we look at an individual window to know that it seems to show very surprising reduction in synonymous rate with respect to both genome-wide averages and also averages that are adjusted for the variation in neutral rates and codon usage bias and composition that you see at 100 kilobase to megabase scale in the genome. So that was an additional filter we did using the same likelihood ratio test approach, which is very important. Okay, so again, we produced a genome-wide annotation of these synonymous constraint elements. I'll now just go ahead and show you a number of really cool examples from these. The first, again, is this HOXA2, which I had started off the talk with. I had shown this lambda s track before, but now we also have these synonymous constraint elements defined at 3 different resolutions or window sizes, 9, 15, and 30. And you can see that indeed those regions where we had observed a reduced synonymous rate are also picked out as statistically significant in these synonymous constraint elements. And sort of both have rediscovered these expression enhancers that have already been experimentally characterized in HOXA2. If you sort of see it in one HOX gene, it's natural to want to be curious about all the other HOX genes. And in fact, it's the case that the great majority of HOX genes contain these synonymous constraint elements. And we're not actually by any means the first to observe these. If you're familiar with some of the initial work with ultra-conserved elements between human and mouse, as well as a few others have observed very striking conservation in the coding regions of HOX genes. So we're not the first to make this observation, but we have genome-wide results here now, and of course the HOX genes are just a subset of that. So there's exciting work going on. Some by collaborators of ours to actually go out and investigate a lot more of these and see do a lot more of them show this sort of expression enhancer activity, or perhaps is there something totally different going on with these HOX genes? So potentially very exciting, again, given how important these HOX genes are to animal development. So here's an example of these synonymous constraint elements within ADAR, the RNA editing gene. So we're looking at the 4th and 5th exons of this gene. The 4th exon you can see is alternatively spliced. So there's 2 different isoforms of that exon, one longer and one shorter, meaning that a portion of the coding sequence of the longer spliced form also encodes the splice donor site of the shorter isoform. Okay? And in fact, we can see evidence for those dual constraints here. So you can see that exactly sort of overlapping where we would expect that splice site to be encoded. We see this reduction in the measured synonymous rate. And then the synonymous constraint elements tell us also that that reduction is significant. And you can see that as we increase the resolution, 30 down to 15 down to 9 codons, we get this sort of pancaking effect where we're zooming in closer and closer on that splice site. This is pretty cool. Here is another example. So this is a gene CEFS2. It encodes something called a selenoprotein, which if you're not familiar with, so selenocysteine is, it's been called the 21st amino acid. In these selenoprotein genes, they're actually encoded by what is normally a stop codon. So UGA, the stop codon, actually in these selenoproteins, the 21st amino acid selenocysteine is inserted in the nascent peptide there instead of translation termination. And that process is directed by a number of elements in the transcript and cellular machinery, but among them, very frequently is a RNA secondary structure, which somehow helps to promote this selenocysteine recoding process. In fact, that's definitely the case for this gene, CEFS2. And we can see here is the stop codon, what would normally be a stop codon, the selenocysteine insertion site, and it's immediately followed by this very well conserved RNA hairpin. And unsurprisingly, as these genes are quite important in various metabolic processes, this gene is, and in particular the selenocysteine residue and the insertion is very well conserved across mammals. Okay, and in fact, we can see evidence for the dual selective constraints on the protein coding sequence and the RNA secondary structure in that region, as indicated by our synonymous constraint element. Okay, yet another cool example. I'll just go through this one quickly. We're looking at two convergent transcripts. Okay, so there's a gene THRA that is transcribed from left to right in this view. And then there's a second gene, NR1D1, which is transcribed from right to left in this gene, in this view. And interestingly, there's a region of the last exon of each of these genes of a portion of their open reading frames that are actually overlapping. So on one strand of the DNA, a portion of the amino acid sequence of THRA is being encoded, and on the anti-parallel strand, the amino acid sequence for NR1D1 is being encoded. And it's actually a quite lengthy region, 66 codons, where this dual coding goes on. So certainly you would not expect synonymous substitutions to be tolerated if both of these amino acid sequences are conserved. And indeed, we see I believe this is one of the top synonymous constraint elements we found throughout the genome, where essentially, again, as you would expect, given what's going on, that region is almost perfectly conserved at the nucleotide level across mammals. Okay, and then the last example in this part of the talk, and this was kind of cool. So this is in BRCA1, which is this very famous gene involved that's been implicated in breast cancer. So we're looking at the beginning portion of I believe this is the longest gene, the longest exon in BRCA1, excuse me. And there's a few interesting things going on at the early part of this exon. So the first thing is it's a protein coding sequence, and the open reading frame starts in some previous exon found much upstream of this view. The second interesting thing is there's a second isoform of the BRCA1 protein that arises from an internal translation initiation. So there's an ATG in the coding sequence of the longer isoform, where under some conditions or at some levels, translation starts at that ATG rather than an upstream ATG, and it's the same frame, but there's an internal translation start site there. And then a third interesting thing, if that wasn't enough, is that there's also apparently an alternative splice form of this exon where there's actually a splice donor somewhere a bit upstream of that ATG. It's in gray here because that splice form is not included in the sort of most conservative human gene annotations, the CCDS set, and looking at what evidence for, there's only like a few ESTs and cDNAs supporting it, which is a little unusual for a gene that's as well studied as BRCA1. I will, however, return to this later in the talk. But what we're seeing now is that there is this synonymous constraint element that sort of captures that internal translation start site, which makes sense if there's some contextual effects in the nucleotide sequence that help to promote this internal translation initiation, which is otherwise a bit unusual in mammalian genes. And we can see our synonymous constraint element sort of captures that exactly. And then we can also look again at the measured synonymous rates and see that there is certainly this measured reduction there. It does seem, although our statistically significant synonymous constraint element does not capture this splice site, it does seem like the reduced synonymous rate extends up to there, but but we can't say that it is significant to call it part of this synonymous constraint element. But again, I'll revisit that later in the talk. But one reason why we were so interested in this example is that it's been a subject of debate actually over the last several years. So this particular region was actually one of the first regions in the human genome where it was observed that there was sort of an unusually low synonymous substitution rate across related species. So that was even before these different mammalian genomes were available. Just by aligning the mRNAs, of course, you can get an idea of the synonymous rates. So back in 2001, it was reported, again, that there was this unusual region in this gene where there was seemingly a reduction in the synonymous rate. At that time, these other splice forms of BRCA1 were not really well characterized or well known. So this paper sort of points out, hey, there's this unusual region here at a resolution of about 100 codons where it seems like there's something unusual going on, but we can only sort of speculate as to what that is. Then a few years later, there was this other paper that sort of revisited that result, and they pointed out some issues with the statistics in the original paper. They brought up some methodological objections there, which were not entirely unfair, but their conclusion was that they thought that this was actually just a spurious result. That they in particular they said it was likely just an artifact of the statistical procedure, or rather that the statistical procedure used in the original paper just hadn't properly controlled for various things that they were doing. And again, those criticisms weren't totally unfair. But now, armed with much more data than either of these studies, we have now sort of revisited this region, and we can not only say that, again, with what I believe to be very rigorous methods, that indeed there is a significant reduction in the synonymous rate here, but now, looking at much more modern and much more complete annotations, we can actually associate sort of very compelling explanations for that observation. Namely, there's this internal translation start site where it completely makes sense that you would have some sort of selection on the synonymous sites in this part of the open reading frame. And again, also that alternatively spliced exon, which is not significant in this analysis, but which I will revisit later. Okay, so this has sort of summed up what is in a paper that is in press in Genome Research on this topic. Unfortunately, because of some delays with the broader paper that is reporting these 29 mammalian genome sequences, it's been sitting in press for several months now, and it will possibly be a few more months before it actually comes out. But I'm obviously anxiously awaiting that all to come together so that the paper can actually come out. But just to summarize so far what's in, and this isn't the end of the talk, I'm going to move on to quickly through a few more things I've been doing since then. But we sort of developed this very rigorous statistical method for identifying these synonymous constraint elements using phylogenetic codon models. We found evidence for more than 10,000 of these located within more than a quarter of all human protein coding genes and covering about 2% of their positions. with resolution between 9 and 30 codons in this paper. And then we investigated a number of hypotheses for what those things are actually doing. Splicing and translation regulation, we saw a couple examples of that. The dual coding genes, that crazy interlocking coding sequence in both strands, those are pretty cool. The RNA secondary structures, we looked at an example with the selenoproteins. And then, of course, these very fascinating tissue-specific developmental enhancers found in Hox genes. And then we had looked at a few other things as well that I didn't get a chance to talk about in this talk, but are in the paper about A to I editing, microRNA target sites, and nucleosome positioning signals. Okay, so that again wraps up what's in that initial paper. I'm going to move on now to a few other results and extensions of the methodology that we have been working on since then. But I guess I'll take a 10-second break here just to take a drink here, and if there are any questions or anything about what we've looked at so far, I'd be happy to talk about that.
It's really fascinating work. One thing I was thinking about though, and this is probably a much smaller effect compared to what you are seeing, but it reminds me a little bit of Josh Plotkin's work on kind of codon usage and the fact that sometimes synonymous mutations aren't actually synonymous because they kind of move you to some place where a further mutation can take you out of that same amino acid. Did you look into that at all? I mean, obviously this is a smaller effect than what you're looking at, but
right. So yeah, the issue of codon usage bias was definitely something we were very concerned about when we were designing this study and thought very hard about throughout the process. So again, the main thing that we didn't control for that was we had again, each synonymous constraint element was sort of shown significant with respect to two different null models. One of which was sort of adjusted for local biases, including codon usage bias, as well as evolutionary, as well as neutral rates and composition. So we performed this test on a little 9 codon window, but this sort of adjusted null model is estimated over the complete open reading frame. So each synonymous constraint element is an exceptional reduction. Excuse me, with respect to, I shouldn't have chosen a fizzy drink, is exceptional both with respect to genome-wide averages and also sort of the average for the open reading frame containing it. Now, to verify that this approach had actually succeeded in sort of excluding regions that are explained by codon usage bias from translational efficiency effects and so forth, you can just very simply measure the codon usage bias in the synonymous constraint elements that come out. And when we did that, in fact, the level of codon usage bias in the synonymous constraint elements is below the average of what you see for genes in general, which is exactly what you would expect if our approach was successfully excluding regions that are in fact explained by this relatively less interesting effect of codon usage bias.
Cool, sounds good. So, I mean, I don't want to cut you off too much, but maybe keep it a little bit short so that.
Yep, I will move through the rest of this pretty quickly. Okay, so the next thing was just a very quick result that you can look at these synonymous constraint results now in HapMap data of SNPs and show that, again, unsurprisingly, they're also under selection in human populations. So this is a cumulative distribution of the minor allele frequency of synonymous SNPs in the Yoruba HapMap population. The red curve is synonymous SNPs inside these synonymous constraint elements, and the black curve is synonymous SNPs found in other protein coding regions. And the fact that this CDF is shifted to the left indicates that when there is variation in human populations in these synonymous sites, it tends to be less than other synonymous sites. suggesting that there is selection on these in human populations, which again, given how well conserved they are throughout mammals, is not very surprising. Next is sort of an extension of the whole approach. What I'm showing here is a pretty amazing alignment. So this is the 5' end of the open reading frame of HOXB5. So you can see the ATG on the left here. And this is, I believe, I think this is the top scoring synonymous constraint element throughout the genome, where it's for actually even more than we're displaying here, it's essentially perfectly conserved across placental mammals. So you can see there's one substitution here. It's a synonymous substitution in guinea pig. That's a low coverage genome sequence, so it's possible that's a sequencing error, or maybe guinea pig has a substitution. But essentially, this whole first I think 40 or 50 amino acids of HOXB5 shows absolute conservation across placental mammals. And again, we're not the first to have observed this. So we're interested. Okay, you go placental mammals, it's perfectly conserved. What if you sort of go out farther in evolutionary time? So I've added onto this alignment now 7 other tetrapod species. So there's some marsupials in here, birds, and amphibians. But sort of very interestingly, right, you can see that the amino acid sequence is still basically conserved, right? Almost all of these bright green substitutions are synonymous, right? But clearly, a number of synonymous substitutions have been tolerated or are observed between placental mammals and other tetrapods. So this sort of suggests the hypothesis that this B5 synonymous constraint element actually evolved in placental mammals. But again, you want to, or we wanted to try to address that in a rigorous way. So we sort of developed an extension of our codon modeling methodology, which sort of builds on ideas sort of widely used with these phylogenetic models, the idea of like the branch test in PAML and so forth. But we figured out a way to formulate a test for reduced synonymous rate, specifically within a certain lineage of our phylogenetic tree. Okay, so just on the left was our previous approach. On the right is very briefly, we now have an additional free parameter, or 2 free parameters for synonymous rates, one within our sort of foreground subtree of interest, and the other within sort of the remainder of our tree. And we can then formulate a test for, do we see a relatively smaller synonymous rate in the foreground than in the background? And then so now re-examining that HOXB5 synonymous constraint elements, when we have a null model, which is the average of all sites in Hox genes, again, that choice of null model is you could argue the pros and cons of that, but that's what we have for now. And so what we see, what we observe by these maximum likelihood estimates is that in the foreground subtree of placental mammals, we have about a 93% reduced synonymous rate compared to the average of these Hox genes. In the background, these 7 other tetrapod species, it's only 47% reduced in that compared to the average amount of synonymous divergence in those tetrapod species in Hox genes. And then again, we have this pretty specific likelihood ratio test, which tells us that in fact we ought to believe that foreground synonymous rate is much lower than the background synonymous rate. The title of the slide remains a question mark because it is interesting that this background synonymous rate in tetrapods is 47% reduced compared to the average of Hox genes, right? So, that's nothing to sneeze at. And this is work in progress, and we're still sort of formulating different ideas about how to interpret that and where to sort of go with that. But This is sort of where we're going in terms of studying lineage-specific synonymous constraint. And then the last thing, which I'll go through very quickly, is we're also interested in increasing the resolution of our analysis. So we had, again, in our initial paper, done 930 to down to 9 codons and so forth. And we're interested, as more species become available, of increasing that resolution even further. But our likelihood ratio test has a few statistical difficulties with this, which are pretty typical of these sorts of frequentist approaches when we try to study a very small sample size, namely the maximum likelihood estimates of our lambda s and lambda n parameters become less reliable. And again, the way that we estimated the p-values for those reductions were based on a large sample theory, which seems precarious when we're, say, studying just 3 or 6 codons or something. So this is not to say that this approach would totally break down. In all likelihood, we could just run it on that sort of window size, and it would probably give reasonable results, but it just becomes somewhat less desirable and somewhat less of an elegant approach when these sort of frequentist assumptions are not something you would want to bet on anymore. So instead, we're developing a Bayesian approach, which doesn't rely on those sorts of assumptions. So forgive the really geeky slide here, but this integral here and stuff is just Bayes' theorem, so don't worry too much about it. But basically, we're going to compute a posterior distribution over lambda s with our our prior and our likelihood term, and we're marginalizing over that nuisance parameter of the non-synonymous rate, and then we'll summarize that with they're with Bayesian confidence interval, Bayesian version of a p-value, and so forth, things like that. So similar approach, but the use of sort of gentle prior information allows us to avoid some of the inherent problems that the maximum likelihood framework runs into when you have very small sample sizes. And then so last slide here, just to revisit again this BRCA1 synonymous constraint element, which was the subject of that previous debate. And remember, there was this alternatively spliced exon here, which had been unresolved because we have, it seems like we had this low measured synonymous rate, but it wasn't included in the statistically significant region for whatever reason. So we're now going to revisit this using more species on the one hand, and this different Bayesian approach for estimating these. So this is like the new track we've generated. It's sort of the odds ratio for reduction, Bayesian odds ratio for the reduction in the synonymous rate, now computed using 33 placental mammals instead of 29. The 4 additional species are primates, so they don't really contribute that much additional information to the analysis. But we're now operating at a resolution of only 3 codons. So we're measuring sort of this evidence for synonymous rate reduction in windows of just 3 codons. The horizontal line here is 15. There's a unit often used for Bayesian odds ratio called decibens. This corresponds to about like 96 or 97% posterior probability, this cutoff of synonymous rate reduction. And so immediately we can see, again, there's this peak, the purplish you can see there indicates that it actually goes off scale here. But so there's a peak corresponding exactly to that internal translation start site, as well as a bit upstream of it. But also now, unlike our previous analysis, we do see a peak corresponding exactly to that alternatively spliced exon. So it seems like now by drilling down further and using a slightly fancier statistical analysis, we're increasing our power and our sort of ability to pick out nearby and perhaps more subtle things like that. And then just to take it even to the next step, we can also compute this with the 40 tetrapod species, so including amphibians and marsupials and birds, and now we're actually just taking it to the extreme. Why not give it a try of measuring this at single codon resolution? We're seeing. I wouldn't sort of go with this analysis just yet, but you can see that again at this cutoff of 97 or 98, something like that percent posterior probability or 15 decibins, we're getting there. We're getting to the point where our 40 species in this analysis is getting to the point where our model is telling us we have pretty strong reason to believe in synonymous rate reduction in those regions. So that's sort of looking towards the future when we'll have even more species available in these alignments. But it's saying with the 40 tetrapods we have now, we're getting there actually. So it probably won't take much more for us to be able to give reliable answers at single codon resolution. Okay, so that was my my seminar for just the anonymous. On the left here are my co-authors on the genome research paper. Manolis Kallis is my advisor. Jacob Peterson and Brian Parker and Stefan did some of the work with RNA structures. Puya did some of the analysis on microRNA binding sites, which again wasn't in my presentation. A number of people on the right here were very helpful while I was doing this work in discussions and advice and suggestions and so forth. We drew very heavily, obviously, on the project that sequenced all these mammalian genomes, as well as ENCODE and the gene annotation subproject of ENCODE and GenCode. And we got, and this was all work funded by NIH and NSF. Okay, so I'll be glad, I guess we're a little bit over time, but I'll be glad to take any questions. But thank you very much for listening in.